good to know that the recording has begun. So yeah, this is the title of my talk. As you can probably guess, it's going to be split into two parts. One where I zoom in to single cell data, neurons, and I'm going to talk about an existing method and some results that I think are fairly practical about it. And then in the second, shorter part, I'm going to zoom out and look at the connectome. And there I'm going to ask a question and then see what kind of math comes out. And it's going to be a little more exploratory in nature. So some early results about a new way of looking at things. Okay. So before I begin, these are the people I worked with on the stuff in my talk. So my supervisors, Heather and Alon at Oxford. The first part of my talk is based on work with Diane and Despoina at King's College London. And the second part of my talk is based on work with Christian and Travis, who I believe was here last year. They started our work together at Oxford, but now both of them have gone off to Texas, and they're both there now. So let's get into it. These are both pictures of neurons. You're likely familiar with them. They're the cells that transmit electrical signals in the brain and nervous system. And as the title of the slide suggests, these both come from Drosophila flies. They come from the surface of larval Drosophila flies, and they're both sensory neurons, so they detect sensation. And they're both different kinds of sensory neurons. And as you can see, they look different. So this one seems to kind of cover the area that it occupies more densely. This one a little less so, but it has all these tiny little spikes, these little protrusions coming out. Yeah. And the other thing about them is they don't just look different, but they actually function differently. So this neuron on the right, the so-called class 4 neuron, it is very good at detecting things like sharp poking, things that the larval Drosophila fly probably doesn't want. And it's not so good at detecting things like a gentle sensation, but this one on the left is better at detecting things like a gentle touch or things like that. So if you decide to pet a larval Drosophila fly, the class 3 neuron is probably more likely to detect that sensation. Okay. So what this suggests is that maybe there's a correlation between the way that a neuron looks, its morphology, and the way that it functions. And indeed, there's plenty of evidence to suggest that this correlation is actually a causal relationship. And so because of that, it's probably important to look at the shape of neurons and study that. So how do I quantify the shape of a neuron? Well, to me, it's just a rooted tree. So I have a bunch of nodes. They represent locations in 2 or 3D on the neuron. And then it's rooted because there's this one particular node called the root that represents the location of the soma. So I'm trying to keep this talk fairly accessible. And so the field that I come from is topological data analysis, which is a very fancy name. But really one of the key goals of this field is to understand geometric data, both quantitatively and qualitatively. So it makes a lot of sense for me to think about this sort of thing. And there's 2 approaches that I want to talk about today about how people use methods in TDA to study neurons. So the first is you directly apply this mysterious algorithm called persistent homology. I'm not going to go into that in too much detail, but this was the approach taken in this paper by Lee and others. And just to go over the basic idea of persistent homology without any math, kind of the vibe of it is that you look at a sequence of shapes. So in this example over here, you start with the stuff that's the furthest from the root of your neuron, then you go a little closer and so on. And then you get this sequence, and then you want to analyze the way that some sort of property evolves through this sequence. In the case of this paper, what they look at is how the connected components evolve through this sequence of shapes. And somehow they want that to say something about the branches. But what I really want to talk about in this first part of my talk is the second approach by Canary and others, including Martina, who might be in the audience. Okay, so I better accurately represent their work. And so in their paper, what they do is they have a much more interpretable descriptor, I would argue, that's inspired by persistent homology, but really is different. and I should say interpretable for the specialized case of neurons. It's really specialized for this application. And so the idea is this. They call their descriptor the topological morphology descriptor, or TMD for short. And the idea is that you give me a rooted tree with some function on the nodes, and that tells me how far each of these locations is in some sense that you choose from the soma, from the root of my tree. And then out comes some sort of canonical decomposition of my neuron into its branches, where I've ripped it apart in some sort of reasonable way. And I think it's best to explain how this works on an example. Here is a rooted tree with a function on the nodes. I pick a branch point. I look at the outgoing branches. I see that the one on top goes out to 3 and 4 microns on its leaves. The one on the bottom goes out to 5 microns. So the one on the bottom goes further out. The rule is I always rip off the stuff that doesn't go as far out. Okay, and then I get this. But I still don't have a collection of intervals. So I pick a branch point. I look at the outgoing branches. One goes to 3, the other goes to 4. After a grueling computation, I deduce that 3 is less than 4, and so I rip off the shorter branch. All right, and there's my collection of intervals, my barcode. Okay, just to make sure I'm crystal clear, another example. Here, as before, I pick a branch point, look at the outgoing branches. One goes to 3 and 4 on its leaves. The other one goes to 4 on its leaves. I have a tie. In this case, it turns out I can choose whichever branch to rip off. It doesn't matter. The answer stays the same. So I'll rip off the top one and then proceed as usual. Pick a branch point, look at the outgoing branches. One goes to 3, the other goes to 4, rip off the shorter one. I'm done. Okay. So in case it's not perfectly clear, here's how you do it in general. You have your tree with your function on the nodes. You pick a branch point, rip off everything except for one branch that goes out the furthest, and there might be this choice. And then if there's any branch points that remain, you go back and choose another branch point and do it again. It turns out it's pretty fast to compute if you start with the branch points that are furthest away from the root and then go towards the root, as Canary and others do in their paper. And again, we will call, because of jargon, the collection of intervals a barcode. Now, one thing that I didn't talk about is how you choose this function. Well, you get to pick, but here's 2 sensible choices, I think. Both of them were suggested in this original paper. So one is the Euclidean distance to the root, which of course gives you some sort of mixture of intrinsic information about the neuron as a metric space and extrinsic information about the way that it's embedded in the real world. But you could also use path distance, which then gives you very detailed intrinsic information, but maybe less additional information about the way it lives in the real world. Okay, so we'll be focusing a little bit on path distance in this talk. Previous work has mostly focused on this, although of course people have used path distance to some extent. Our first result is something that was kind of alluded to in earlier work, but there wasn't really a proof of it. And it wasn't so difficult, but it's basically that if my function is increasing as I walk away from the root, it turns out that the TMD barcode that I get is the same as the barcode, or as a particular kind of barcode I could get from this persistent homology approach that Lee et al. Use. And so from this, I have a couple consequences. First of all, if I'm doing path distance, that always increases as I walk away from the root. So of course, then this TMD is actually under the hood just a persistent homology calculation. Okay, and if it's Euclidean distance, and then it happens to be increasing along paths away from the root, then it turns out that by applying a result from this paper by Lee et al., this descriptor records what's called a neuron's Sholl function. I meant to get a whiteboard out, but I'll just explain this with my words. Sholl function is a very classical descriptor, just in case we're not all familiar. The idea is you give the Sholl function some positive radius. You look at the sphere of that radius around the soma, and then it returns the number of times that the branches of the neuron intersect that sphere. Okay, so this is a very classical descriptor, and the idea is just that it records kind of the branchiness of the neuron at different scales away from the root. Okay. But so there's some issues with barcodes, and these are common issues in TDA that there are many different approaches for. So I'm just going to discuss, well, first let me say what the issues are. So this is a barcode from a real neuron, and this is from the paper of Canary and others. And I think personally that is a little hard to interpret kind of what's going on just because there's so many intervals there. But there's some other more technical issues, like the metrics on the space of barcodes can be kind of complicated for doing data analysis. So, for example, I don't really know what it, or, there's not always a good way of saying what it means to take an average of a collection of barcodes. What does it mean to subtract one barcode from another? I don't really have an answer to these questions. Of course, we need to address these, and Canary et al. Do, and I'm going to, in almost the same way they do, but with one slight modification that's very important, and which I will highlight. So the first idea is just for the interpretation. What we're going to do is we're going to kind of condense this information into a collection of points in the plane. So what you do is, so I have this interval from 2 to 3. It has left endpoint 2 and length 1. So I send it to 2, 1. This interval from 1 to 3 has left endpoint 1 and length 2. So I send it to 1, 2, and so on. And then this gives me a collection of points in the plane, which I call a transformed persistence diagram. And you'll see that the X and Y axes are labeled by birth and persistence. And this is also just the jargon of the field. Okay. So I would argue that this helps with this interpretation question, but it doesn't really help with my more technical issues. So what I'm going to do is I'm going to turn this into a vector somehow. And I'm going to do that by imagining that there's a Gaussian hovering ominously over each point in the plane. And then when I add these up, this will give me a function that I call a persistence surface. And then this function is a vector, and I can average and subtract these functions very easily. And this is an idea from Adams and others. Now, one thing that you'll notice that I do, that Adams and others do, and Canary and others do not do, is that I have been really unfair with my Gaussians over here. So I've weighted them by how high their corresponding points are above the x-axis. So this one's way higher up than this one. And this is the crucial difference. Okay. And then lastly, what I do is I sample this function over a grid, and this gives me a matrix called a persistence image. That's just a list of numbers. It's a vector. I can take averages of it and all that. Okay. And there's also an interpretation for these persistence images. So if I'm using path length for my TMD, then warm regions up top correspond to long branches. Warm regions in the bottom correspond to short branches. Warm regions with large X correspond to branches that initiate far from the soma. Warm regions with small x correspond to branches that initiate close to the soma. And then because of my weighting, although this isn't why I'm doing the weighting, it just so happens that the net intensity of my persistence image is proportional to the length of the neuron. And of course, this admits these L1, L2, L infinity metrics and lives in a vector space and all that. So I would say that the problems are solved. Okay, so another result, I guess. So then, since we were using path length, we thought, what is the path length analog of a Sholl function? Well, so we just called it a topological morphology function. And the idea is that you give it some positive number s, and then it spits back the number of points that are path length s from the soma. And people have, of course, thought about this notion before. But I just want to point out that, of course, and in fact, it's not so hard to show. You can recover this from the barcode and indeed from this transformed persistence diagram. So from the barcode, what you would do is you would count the number of intervals containing S and then look for that. And that corresponds in the world of these transformed persistence diagrams into counting points in triangular regions. Okay. So that's pretty straightforward. Okay, then what we showed is that you can actually also approximate this from persistent surfaces and images. So in fact, if instead of counting points in these triangular regions, I do a weighted integral over this region, then that weighted integral will give me a smooth approximation of my topological morphology function, and it will converge as the standard deviation of my Gaussians goes to 0. And this also wasn't so complicated. Okay. Now this is why I weighted the Gaussians. Okay. And it's because we showed that these persistence images, which people are using to analyze neuronal data, are stable under two kinds of perturbations. So the first is I can perturb my function on the nodes. And the second is I can add or remove a short branch from my neuron. Now, I want to be very clear about two things here. So the first is I'm not claiming that I can just add as many edges as I want to my neuron. And in fact, that's not going to be stable. Okay. And indeed, I might actually not want this to be stable. So remember this neuron from earlier at the beginning of the talk that was actually characterized by having lots of short branches. So this, depending on your application, you may not want this kind of stability, but you may. This is how it is anyways. The other thing that I want to do is put this into context with related work. So there's already this result from this Canary paper again, that actually the TMD algorithm is stable for barcodes, but not for persistence images under this metric called the bottleneck metric, which people in TDA will no doubt be familiar with. So the problem with this is that it doesn't imply the stability of persistence images. So we instead showed that these, on a technical level, these persistence or these barcodes are stable under what's called the one Wasserstein metric. And this stability does imply stability of persistence images. Now, I should add that I actually expect that the first kind of perturbation is also bottleneck stable, although this isn't something, I expect that persistence images that are unweighted are actually stable against the first kind of perturbation, although I didn't prove this. The other thing is that this first kind of stability isn't so hard to show from bottleneck stability. So the main contribution that we have is the second kind of stability. And indeed, this first kind of stability has already been studied a little bit for specific kinds of functions. Okay. And then we looked at some data. So we looked at what are called tau35 mice. So these are mice that have been genetically modified so that they have an accumulation of toxic tau during their lifetime. And this causes their cognitive ability to be impaired in roughly the way that you'd expect for someone with a accumulation of tau, pathological accumulation of tau in the brain. And so there were a few variables that our neurons had. So they differed by strain. Some of them were tau, some of them were wild type. We had some at this pre-symptomatic 4-month age where these tau35 mice would not show any symptoms really. And then this post-symptomatic age at 10 to 12 months where you would really notice if the mouse was a tau35 mouse or not. And so we were wondering if fixing the other variables, these two kinds of neurons were on average different. Okay, and so we did a simple randomization test. We fixed an age and a region, and then we computed the L1 distance between the average tau35 group and the average wild type group, right? So we took all of their persistence images using these 2D projected path length. And then when we took the average and took the distance, we then compared that to when we did the same thing, but we randomly shuffled the groups 10,000 times. And we saw the percent of times that the randomly shuffled groups had a distance less than that of the true groups. And what you see is that most of the time that was the case. So we suspect that there was a significant difference between the two groups. Well, especially, or at least for the 10 to 12 months, the post-symptomatic age. So then we looked at the persistence images. Okay, so here's the average wild type persistence image. Here's the tau35 persistence image. And here is this one minus this one. And so as you can see, there's more of a warm region up and to the right, suggesting basically what you'd expect, that the branches in the healthy mice are both longer and initiate further from the root than in the diseased mice. And we see the same thing in the hippocampal neurons. Okay, so that's part one. Oh my God. Okay. So part two is motivated by this question. So if I have some connectome representing the brain, and I have some model of a toxic protein on my connectome, then can I use purely topological features, and actually topological features, and I mean this in a fairly coarse sense that I'll define later, can I use topological features to characterize the spread of toxic protein, or do they not matter? Of course, just for anybody who doesn't study this, it's fairly simple. The idea is that I would seed the toxic protein at some node, and then I'd let it diffuse throughout the graph, and then via some model, I'll say that after some time, eventually an edge gets damaged, then another edge, and then eventually, after some time, all of the edges are damaged in my graph. Fairly straightforward. And so then I get a sequence of damaged edges. And I can use this to make a sequence of graphs by starting with every node, and then I gradually add edges. And so if I were to do this persistent homology thing, and maybe you know what that is, maybe you don't, well, that asks how homology evolves through this sequence of graphs. Now, you don't need to know what homology means because I'm not going to ask that. Instead, I'm going to ask how the homotopy type of these graphs evolve. And I'm just going to see what mathematically comes out of that. Now, you might not know what homotopy type is, so I better explain that. So this is a graph, I think you'll all agree. And it has 4 connected components. And each of these graphs, or each of these connected components, has some number of loops, okay? And that's all that the homotopy type of my graph is. It's the knowledge of each connected component and then the number of loops that each of these connected components has, okay? So if I want to be a little more succinct about representing this, and I do, then what I could do is I could represent each of these connected components by a monomial, where I've just had the exponent tell me the number of loops that that connected component has, okay? And then if I wanted to be even more succinct, I could make a polynomial. And then that polynomial tells me the number, or the homotopy type of my graph. So here, just by looking at the polynomial at the end, I can tell that there's 1 connected component with no loops, 2 with 1 loop, and 1 with 2 loops. And this is just a notational convenience. There's nothing mathematically deep happening here. Okay. So then going back to my sequence of damaged edges, I get a sequence of polynomials that tells me what's happening up to homotopy. And so this lives in a kind of complicated mathematical structure called a graded poset. And I named this structure fancy H of n. And you might not know what a graded poset is. So let me just tell you how I construct fancy H of n. And I'll show you on this example, fancy H of 5. So the idea is I've written down a bunch of polynomials. Okay, and in fact, I've written down a polynomial representing every possible homotopy type that I could get on a graph of 5 nodes. Okay, and here they all are. And then you wonder, why have I drawn arrows? The idea is that there's an arrow from one homotopy type to another if I can get from one homotopy type to another by attaching an edge. Okay, so then this partial order thing, all that means is that one homotopy type, I would say, is subordinate to another if there's a path from it to the other homotopy type. So this, the type represented by 4 is subordinate to the type represented by 2. Okay. Then there's this notion of a grading. So as you can see, I have these vertical strips in this picture, and these are what are called the gradings. The idea is that if I look at each vertical strip, I can see how many homotopy types there are on, say, in this example, 5 nodes and a certain number of edges. So for example, these 2 homotopy types are the only homotopy types I can get with 5 nodes and 0, 1, 2, 3 edges. Okay. And then finally, we were looking at these sequences of polynomials And so these I can extend to just be maximal chains in this thing. Now, what's the maximal chain? That's just a path that starts at the start and ends at the end. Okay, so really the idea is that I've made this structure, and then any sort of sequence turns into a path from the start to the end. Fair enough. Okay, so then just as a simple proof of concept, we ran a very simple model of Alzheimer's disease, no even amyloid beta thing in there. And the idea was we just simply perturbed it by the seeding location to represent different subtypes of the disease. And what we saw was that, so even though we were using this fairly coarse description of the model, we saw that actually 3 out of 4 of our subtypes differed by their homotopy type. Okay. So then we thought about it.
Can you explain the figures, the results a little bit more?
Yes. So, I mean, basically, really, I mean, what we did was quite simple. We looked at the paths, and then we saw that they were different. And so, and in fact, and I forget which ones, but like what we saw was that some of them tended, I think, slightly more to aggregate, or tended to, you know, basically there was this. Some of them tended to form more loops, whereas others tended to simply branch out in sort of like a tree structure at the beginning, I think is the difference. So, yeah.
But here you have this some super dense. Poset here, this blue one, and then you have this
so
paths in the poset. So what happens when you go like
so one sort of unfortunate thing is that we couldn't think of a canonical way to sort of order the nodes in and so these. So I should add these blue graphs. These blue graphs represent these fancy H's. Okay, and so one sort of trouble we had is that we couldn't figure out sort of a canonical way to vertically order the nodes. So then we started thinking about this is a metric space. So we looked at all the different paths and we wanted to say how different are they. And so what we said was that these maximal chains, so these paths from the start to the end, are adjacent if they differ at one point in the grading. So here, if I take this red path on the bottom and this red path on the top, these two are adjacent because they only differ in this layer. Okay, and then if I take a sequence of graphs starting from, or a sequence of paths starting from one and ending at another, so starting at F and ending at G, and then this sequence, the neighbors are always adjacent in this sense I defined above, then I'd say that if I have a sequence of length D, then I would say that there is a discrete homotopy of length D between 2 maximal chains. And then finally, we defined this notion of discrete homotopy distance is just the shortest length I can possibly obtain of a discrete homotopy. Okay, so one issue is that in an arbitrary poset, and maybe you don't know what that means, but indeed in this fancy H of n structure, why should I know that a discrete homotopy always exists? Well, this was something that we had to prove, and so we showed that it's true. And so indeed, this means that the discrete homotopy distance is a metric on the space of maximal chains. So there's an open problem here, which is that the number of maximal chains is very large. And let's see how I'm doing out. Oh, I have plenty of time, actually. Cool. So more on this in a second. And so directly searching through the space of maximal chains is quite slow. And so we'd like a better way to compute this. And this is something that we're thinking about now. And if not to compute it, then do approximate it. So, okay, now I always seem to go too fast in these talks. And so it means that I always leave a little something in case I need to spend some more time. So I'm going to go on a bit of a combinatorial digression on the size of this fancy H event. Okay, so this is bonus content. So before I can get to that, I need to talk about this number CK, which is the minimum number of edges I need to get a graph with K loops. Okay, and if I have a connected graph I can always, or a connected graph that isn't complete, I can always just add an edge and then I get another loop. Okay, so I've done that up top. But if I have a complete graph, so this triangle here, and then I have a spare node lying around, I can't just add an edge inside this complete graph because it's complete. So I have to use a spare node and 2 edges to get another loop. Okay? And so then you have this heinous equation that tells you the number of edges you need to get k loops. But the idea of this equation is fairly simple and is brought about by this observation here, which is just that this sequence of CKs skips over numbers of edges on complete graphs plus 1. And this turns out to be the right thing to do. I don't want to go into the details of that. I just want to say what this means combinatorically. So, okay, so remember that the size of these gratings, these vertical layers in my fancy H of n is the number of graphs up to homotopy with n vertices and m edges. Okay, so equivalently, I could ask how many graphs have m edges and Euler characteristic L. So the Euler characteristic is the number of nodes minus edges. So again, equivalently, there's this bizarre game I can play where I can basically act like I'm going to the connected component shop and I have some allotment of edges and I need to reach a target Euler characteristic. So I buy selections from this list of pairs. Okay, and I'm not allowed to exceed my price of edges. Okay, and so I'll call this number of ways of homotopy types on m nodes and or m edges and with Euler characteristic L, I'll call that big H of m and L. Okay. So one sort of irritation of this reformulation is that I'm getting equal to L, but I need to stay under my budget of M. So this is a little inconvenient to work with. So what I do is I add another row, and this extra row is saying I don't change my Euler characteristic at all, but I can spend the rest of my money on this useless item. So by adding this, then I can instead ask how many ways can I buy items from this new table so that I spend exactly m edges and get exactly an Euler characteristic of L. As a minor detail, sometimes by doing this adjustment, I have to subtract 1 because of an issue involving the empty graph. It doesn't matter. So what I'm going to do is I'm going to compare this to a seemingly easier problem. So how many ways can, let's see how I'm doing on time. Yeah. How many collections of items could I buy from this list, spending exactly a number of, I don't know, n dollars, I guess. So I have one number, so I have every positive whole number in my list. And this seems like a pretty innocuous problem, but this is actually a really famous and difficult problem. So this is the number of partitions of n, and has been studied by a bunch of number theorists who put a lot of work into this. So it's actually, even though I'm saying this very condescendingly, it's actually quite a difficult counting problem. So and this is the easier problem, by the way. So here's a problem of intermediate difficulty. How many collections of items could I buy from this new list where I've skipped over some numbers that I decided I didn't care about? They were out of stock today. And how many ways could I buy from this set of prices, spending exactly n dollars, I guess, since I'm American. So there's this theorem that says that if the GCD of these numbers is 1, and this is also from a number theorist, and these numbers have a positive natural density, then the log of this grows like n to the 1/2, basically. Okay. So, and then we have all these crazy constants. Okay. So we exploited this like crazy, this connection. So first of all, it actually wasn't so hard to show that this number is weakly increasing in this direction. So if it goes up if I go this way, and it also goes up if I go this way, with one exception at the origin. Secondly, we had this really nice reduction that if I go up enough, it stops to change in the up direction. So once I cross the x-axis, it's no longer increasing anymore. It just stays constant. And actually, it stays constant in terms of one of these intermediate problems. So if I want to compute h of m and L, where L is positive, I decided to call that number h of m. Okay, because it no longer changes in L once L is positive. And so what we realized is that this h of m is just one of these intermediate problems. So there's this set where I just need to find out how many ways I can add up numbers from this set to get this value. Okay, and then as a result, you get this upper bound on the size of h of m and L. Okay, so from this, it's not too hard to show, but I think I'll skip the details, that actually, just by combining these results, that in fact there exists, that the log size of this fancy h of n grows really fast, or it grows reasonably fast. It grows between the square root of n and linearly in n. And I don't know what the details of that are, something I'm kind of interested in. What this means is that when I don't take the log, it grows very fast. So this is justifying that we'd really need to think of something better than direct search. But also I think it's just interesting in its own right. Again, this is all bonus content. I didn't intend to talk about it. Let's see. And the same result holds when I replace this size of fancy h of n with the size of its largest grading. Okay, so going back, let's see. So back in the first half, we looked at this TMD thing. We saw that it can capture joint information about branch length and branching distance. We saw that it coincides with persistent homology, this nebulous algorithm I never defined in certain cases. It encodes a path length variant of a neuron's Sholl function, and persistence images encode an approximation of this function. And then persistence images arising from this TMD construction are stable and interpretable if you do this Gaussian weighting at least. And we then use persistence images to describe differences between wild type and diseased neurons. So then finally, in the last part, we looked at a framework for analyzing homotopy types of sequences of graphs with an eye towards seeing how this can or cannot characterize how disease spreads. And then we provide some asymptotic bounds on some combinatorics. Although when I did a practice talk, I went at a slower rate and didn't think I'd get to that. Okay. Yep.
Questions, Yanis?
So with these maximal chains,
Yep.
did you do you have real data to compare it to see to measure the distances between what happens or something simulated?
Yeah, so we just did it on simulations.
Okay.
So yeah, so I mean, really, this was kind of like a first pass, and a lot of it was a methods paper, but you know, with an eye towards this question.
And was there do you consider the time component, like maybe it goes along the same path, but it just takes slower or faster?
No, I think this is definitely like a reasonable addition, and I've been thinking about this a little bit, just in my mind, not like, yeah. So because, in fact, if you wanted this metric to be stable, then you'd have to incorporate the time component. Otherwise, it's a purely combinatorial metric. But I'm thinking it might be able to perturb it in some way. And in fact, if you include the time component, it would be, this is how you would relate it to more classical metrics that people use in TDA. I think that this is definitely a relevant thing to mention. Yep.
Did you look at the structure of the poster? Is it a lattice? Because it looked like the ones you showed all looked quite latticey.
I didn't even think about that, to be honest.
And then I was wondering if you looked at the order complex of that poster and the homotopy type of that, just because then you're looking at homotopy types and homotopy types.
Oh, yeah.
Niceness to that.
No, that would, I mean, I think there's like a lot of really interesting things to do with this poset. Like you could, I mean, as you can see, I went in like some ridiculous pure math direction with it. And like, I think there's a lot of like interesting structure there. So yeah. But I haven't.
Yeah. So regarding this functional summary you define for the TMD,
Yep.
so I think the Sholl we have this terminology Betty curve for the
Yep.
barcode. I think the Sholl function is the Betty curve for the Euclidean distance, and if you compute the barcode for Euclidean distance.
Yeah. So if you computed the barcode
The
with
TMD
with the
Euclidean
TMD,
distance.
I guess if the function was, if it was increasing as you walked away from the root, then yes, I think so. Yes.
Yeah, but I wanted to actually
Yeah, no,
go in another direction. Sorry, I just got mixed up. But this function you define,
Yep.
I don't think it's very stable. Do you have stability results on that?
The path length one.
Yes. I think it's the Betty curve of a barcode.
I
So
think.
don't expect it
Okay.
to be very
Well,
stable.
now I have to think on the spot. I'm pretty sure this one actually is stable. So the Sholl function, I could imagine being unstable.
Mhm.
But I think this one is, because let's see, if I added a short branch and it's dependent on the length, I think this one is stable, I believe. Certainly to adding a branch, and also I would believe to perturbing I don't know why I've landed on this slide.
But
I think so.
stability with respect to the usual metrics we use in persistence, for example.
So stable against perturbing the barcode, you mean? Yeah, that's a good question. Well, I guess like it's certainly dependent on the number of intervals you have. But if you're allowing me to depend on the number of intervals, I would think it is stable, but you know.
Maybe respect to Wasserstein, not
Yeah.
Okay.
Yeah, exactly. Actually, that's probably how it is. Yep.
I have a quick question. So your H of N, I forget the name.
Yeah.
Your poset.
Yeah.
You said it's huge and you cannot look for all paths.
Yeah.
But when you construct it.
Yeah.
You're adding edges anywhere in the graph, right? And if this would be the spread of disease, you would want to add an edge only if it's neighbor to already a contaminated edge.
Yeah.
Right, so you will want the set of edges to be connected, and then you might get a much smaller poset than this poset.
Yeah. So in and in fact, so this is a good point. And actually, so in our revision for this paper, we're actually going to look at the instead of doing this path up, we're actually going to look at the path down, where you look at the healthy edges. And the reason is because actually in this case, you would expect, for the exact reason you just said, you would expect for the sequence to be more interesting in terms of homotopy type. And indeed, yes, in fact, in this case, it is actually not so bad to compute because of this.
And from the biology part of it, is there any reason to think that looking at cycles at each edge count is somehow telling you something about disease?
Yeah, so the reason that I got involved in this project was because Christian and Travis were talking, and actually neither of them are in TDA, but they wanted to know if they have this model, does it tend to form connections within, do the disease tend to form connections within the already diseased network? Or does it tend to branch out into, you see? So this is what they were sort of interested in. But then they, that was kind of how it started. Yep.
I just think this distance, if you consider your process as an order complex, then
Yep.
the discrete homotopy distance
Yep.
is the same as the Q connectedness distance through your order complex.
Could be. I don't know what those words mean.
Yeah.
That we should talk later.
Yeah.
Great.
I think let's thank David now. And continue in 3 minutes with Mita.